This is Dr. Sargisian, and it's our Genes and Genetic Diseases lecture. And I'd like to have a short review part for you, just to touch base on the subjects that we are going to talk about and what's the basis for those subjects. So definitions for the general terms are given here, and we need to remember that genetics is study of inheritance, of biologic heredity, and gene is the basic unit of heredity. Don't confuse it with chromosomes as a unit of heredity or codons. Gene is the basic unit of heredity, and by basic unit of heredity we mean that gene has that sufficient amount of information to transfer or to pass one or the other thread, and the traits will be passed based on genetic composition, based on genes. The codons alone don't have enough information, and chromosomes have several or lots of genes. So, but to pass one trait, you need just a gene. Genomics is the field of genetics that studies the structure of the genome, and genome is the DNA that represents all the genes for a given type of species. So again, genome will be representing everything, all types of genes you can imagine for this type of species. So of course genomics and genetics not only pertaining to human physiology, human pathophysiology, but also animal and plant or even microbial world. Everything controlled by genomes, genetics, and genes carry the information in these types of living structures. When we're talking about heredity, we need to talk about DNA and genes. Main point here that genes are the sections or sequences of DNA that have the instructions to make a single protein. Of course, there can be lots and lots of genes in one DNA strand. We are talking about bases, and bases are adenine, thymine, guanine, and cytosine. When we are talking about DNA, RNA consists of similar bases, however with one difference. In RNA, the thymine is replaced by uracil. All ANP pathophysiology biology instructors love this difference and may include hint it in the test. The amino acids are strung together and they attach together to make a protein. Protein is polypeptide chain, so amino acid will be the peptide. So basically DNA presents a coding for future protein. DNA are located, it's of course in the nucleus, and DNA has a double-stranded structure which somewhat meaningless. I'm talking meaningless because one of the strands has no function at all. The function of the meaningless strand is complementary. The main strand, we can call it meaningful, carries the genetic information, while the meaningless strand is complementing the meaningful strand. So basically the main strand is complemented with meaningless strand in order to give structure and, I would say, sturdiness to DNA. However, when it replicates, it creates two identical molecules of DNA, and you can see on this picture how it happens. So meaningful strand attaches to another new meaningless strand, and meaningless strand attaches to a new meaningful strand. Transcription occurs inside of the nucleus, and transcription is the initial step to get the genetic information out of the nucleus and move it to cytoplasm for the creation and construction of the proteins. In what type of cells this can occur? Only eukaryotics. Important concept, eukaryotic cells, and I'm sure you all know already that eukaryotic cells are the cells that have nucleus, and nucleus contains DNA. Prokaryotic cells don't have nucleus, so that's the main difference. Of course, human cells are all eukaryotics. So messenger DNA creates a copy, or it's a copy of meaningful strand of DNA. Actually, it's a complementary strand to DNA, and eventually it moves to cytoplasm. The main thing in this picture I would like for you to pay attention, it's the stop codon. It's when the transcription ends. The stop codon tells to end the transcription. However, before the messenger RNA moves into cytoplasm, some changes have to occur. As you can see here, there are exons and introns. And introns, we can spend about 24 days discussing them. However, in this case and for our course, we will treat them like useless, meaningless particles that connect the exons which carry the information. So the process called splicing, and splicing occurs inside of nucleus. Then the RNA, messenger RNA, is reassembled and sent out cytoplasm. When it seeks the ribosome, ribosome is, as you know, it's the assembling line or the plant where protein synthesis occurs. So messenger RNA will attach to ribosomal RNA and start running through the ribosome. At the same time, amino acids are carried into the ribosome by transfer RNA. Little tracks, they carry the amino acids, and the whole process leads to this creating a chain of protein or polypeptide chain. So everything works like assembly line. There are 20 amino acids, and there are 20 different types of transfer RNAs. So all together they work and assemble the protein. Of course, in this case, just assembling the protein, it's not enough. The protein has to be folded and transported to an appropriate place in the cells. And the folding occurs with the help of these particles which are called molecular chaperones. And chaperone in this case, that's someone that oversees the protein and controls the behavior. So protein won't unfold and will stay in the folded state and act as needed. So those are molecular chaperones. However, it's possible that eventually the protein may unfold and it may contribute and attach to another protein, which unfolding, we all know the process is denaturation, and attaching and unfolding of the protein can lead to many diseases, and the example may be the Alzheimer's disease. So ribosomal RNA, again, just to review that it moves along the messenger RNAs and again moves the transfer RNAs into position. But you have to remember this, that even though this is a teamwork of 3 different RNAs, the only RNA that actually goes into nucleus is messenger RNA. Everything else is in cytoplasm. So ribosomal RNA, again, just to review, that it moves along the messenger RNAs and again moves the transfer RNAs into position. But you have to remember this, that even though this is a teamwork of 3 different RNAs, the only RNA that actually goes into nucleus is messenger RNA. Everything else is in cytoplasm. So when the cell divides, it needs the duplication of DNA. As we know, the DNA is folded into chromosome, and ends of chromosomes are telomeres. The telomeres used for attachment of the enzymes that facilitate the duplication of DNA. However, the end of the telomere does not get duplicated, and eventually telomeres wear off. We talked in the past about aging theory, that wear of the telomeres causes aging, and this is a question for you in this slide, that a man with a mutation that causes his cell to rebuild their telomeres after every division, and what will happen with this man. Is it a good thing or bad thing? I think you know the answer, but you can think about it and include into your discussion or your thought process whatever we talked about before in previous classes. I'd like to talk about meiosis and mitosis, and mainly will focus on meiosis. Mitosis, you recall, that is a regular cell division which we need for replenishment of some wear and tear, such as blood cells. We need to make new ones, or skin cells. So basically regular skin division. However, meiosis leads to creation or production of gametes or sexual cells, or in this case sperm and ova. So just very briefly, that in meiosis the chromosomes duplicate, and one pair will go to each daughter cell on the first division. However, one chromosome of each pair will go to the daughter cell on the second division. I'm talking about it because this will be very relevant about when we talk about genetic disorders. And one of the phenomena we want to touch base here is crossing over. When chromosome pairs are lining up during meiosis, they may and they will exchange ends. So they will have the resulting gametes or sexual cells, and when we're saying gametes, it's really no difference from this standpoint. Is it sperm or ova? They will have different combination of the genes, and here you can see like the tips are exchanged, so which results in richer, broader genetic variety. So another phenomenon is linkage. When 2 genes are standing close together on the chromosome, are linked, they will be very unseparatable, or it's very difficult to separate them by crossing over, and those genes are usually inherited together. So very, very brief review. So when we're talking about inheritance, we need to talk about Mendel's law, and I just took this picture from the book and Punnett square the genetic inheritance process. You see the probability of inheriting each trait, and capital D means dominant, small d means recessive. So, and you see like ovum and sperm interact and the resulting cell, what it will have. Just for review, if two dominant traits interact together, create a new cell, that will result in dominant traits. Dominant trait with recessive trait results in dominant trait, and two recessive traits result in recessive trait, and everything is in this little square. So of course it's much harder to develop recessive trait. You need two recessive traits supplied from each parent, and it's much easier to develop the dominant trait. So if you think about the square here, the recessive trait occurrence will occur only in 25% of 25%, of cases. So when one of the phenomena when we're talking about inbreeding or close of marriage between close relatives, what happens, the supply of recessive material, recessive genes increases, and that's why there's a concern about that type of relation, because recessive genes sometimes are related to diseases. That's why the offsprings of marriage between close family members may have some health problem, some difficulties, may have cognitive or physical health problems. So alleles, it's a copies of a gene. You know, if the copies of the gene are alike, it's homozygous. If different, heterozygous. And again, if you are heterozygous for recessive trait, but anyway, you don't show the trait because it's recessive, you are a carrier. If you have only one copy of the gene, you are hemizygous. Just simple definitions. Again, about carrier, basically the gene isn't showing up, it's not expressed, and only the notion of the carrier or concept of the carrier can only relate it to recessive genes, right? Because if it's dominant, it will show up. Okay. Genotype versus phenotype, very easy. Genotype, it's the genetic material, it's the blueprint how to assemble the person, basically, and phenotype, the actual physical characteristics. So easy way to explain it, if person has, and the book brings the example of the eyes, and blue eyes is recessive and brown eyes are dominant. So person with brown eyes may have genotype of two genes supplied from mother and father, both dominant, or he or she may have a genotype of one set of blue eyes, recessive gene, from one parent, mother, and brown eyes from one parent, father. So, and again, the person will have brown eyes. So person with brown eyes may be carrier of a recessive gene of blue eyes. So, and I left this discussion on this slide, so think about it, and I already gave you the answer. So gene expression. So gene expression related to the fact that genes are actually turned on or turned off, and induction and repression. Induction, there should be some kind of trigger to start a gene, to turn a gene, like it can be external factor, or repression, something turns the gene off. So is the protein is actually produced, the code it's standing for. And if gene is turned off, of course the protein is not produced. I'd like to talk about gene expressivity, and it's very easy concept to understand. Means it's expressed when it shows up. So here we are talking about if the protein is made, does it change person phenotype, how that person looks, how his physical appearance or physical composition, chemical composition different. So no, we can't see it, so we will call it recessive. The gene will be called recessive. Yes, and we will call the gene dominant. And if it shows up sometimes that it has intermediate penetrance. Let's talk about gene interactions, and this is a list of terms, and I just would like to go over them and give you these definitions. So if a person exhibits a trait and the trait is secondary to many genes, we will call it polygenic. If a trait is expressed secondary to genes and also the environment can affect it, then we have multifactorial expression. So multifactorial is a good word because not only in genetics but everywhere else, if several factors influence something, you definitely can use word multifactorial. If you and most of the events, of course, are multifactorial. Very rarely events can be secondary to single factor. When one gene masks the effect of another one, that epistasis, basically, if you have one gene, the other one never will express. And when genes depend on each other, when they work collaboratively, we talk about complementary work. Two genes together may work and create a new phenotype, and we call this collaborative. The difference between complementary and collaborative is when we working with complementary type, it's when two genes depend on another. They cannot work separately. When it's collaborative, we can say that they work together to create a new phenotype. However, they probably could have create it alone. So, and this will be our short list of definitions about gene interactions.